All right. I think I will skip over the basic logistics and just go straight into our announcements. So next week's seminar on Thursday, October 20th at 3:30, Karen Barcelo with CIMERS, which is the Cooperative Institute for Marine Ecosystem Resource Studies, will be talking about integrating spatial population structure and ecosystem information into stock assessment. Please join us in person here in the Gladys Valley Marine Studies Building Auditorium or online. If you need info or login details for this or any of our events, please log on to the HMSC homepage, scroll to the bottom for a calendar of events with all the details. With that, I'd like to introduce our seminar host, Dr. Scott Baker, who is the Associate Director of the Marine Mammal Institute and also professor in OSU's Fisheries, Wildlife, and Conservation Sciences, otherwise known as FWICS, and is also an adjunct professor in the School of Biological Sciences at the University of Auckland in New Zealand. With that, Scott.
Thank you, Ichang. So it's a real pleasure today to introduce Dr. Keith Hernadez as a recent appointment as a postdoctoral scholar in my laboratory here at the Marine Mammal Institute. As I think you'll see today, Keith is broadly trained as a population biologist and molecular ecologist, and has worked on topics ranging from acoustic ecology of fish to pinniped demography. Here at OSU, Keith is developing and applying epigenetic clocks for determining the age of the critically endangered Māui dolphins of Aotearoa, New Zealand, and the endangered Cook Inlet beluga whales of Alaska. He finished his bachelor's in marine science with honors from the University of Hawaii in Manoa. Hilo. I went to the University of Hawaii in Manoa. Old rivalry there. He finished his master's in marine science at Moss Landing Laboratory, working with one of our old colleagues, Jim Harvey, and he completed his PhD at Louisiana State University. And with that, I think I will turn it over to Keith to tell us about from pinniped diets to aging dolphins, one postdoc's path in marine ecology so far.
Cool. Thank you so much, Scott, for that introduction. And yeah, let's dive into some science. Hope if I'm not speaking loud enough, please let me know. There we go. Cool. So top predators are critical components of the ecosystems that they inhabit. We are probably fairly familiar with the idea that they exert top-down forcing on lower trophic level organisms in the ecosystems they inhabit through predation, but they can also do this by exerting competition or ecology of fear on lower trophic level predators. They're also really useful to study because they act as sentinels of ecosystem health. We know this through studies of their diet, which give an indication of the prey communities that they interact with, but also studies of their tissues gives us an indication of their exposure to contaminants or other organic pollutants. These top predators also tend to interact with humans in a variety of ways, and we'll talk about some of those in this talk. So many populations and species of marine mammals were exploited in history for harvests, such as through commercial whaling or the fur trade for seals and other marine mammals. And many of these populations have since recovered from this historical exploitation as a result of implementation of laws such as the Marine Mammal Protection Act or the Endangered Species Act. But as these populations are recovering and returning to places that they were historically, they're also coming into more frequent contact with humans through a variety of activities, one of which is the fishing industry. And these interactions with fisheries can be negative for both marine mammals and the fisheries that they interact with. Marine mammals can depredate catch, they can also damage gear, they can also accidentally drown in gear or maybe intentionally killed on occasion. And so it's necessary for better management of marine mammals as well as the fisheries they interact with to understand how these interactions are occurring. And we'll focus on some marine mammals today, mostly the pinnipeds. So these are your seals and sea lions, and they're globally distributed, as you can see in this map here, with many species found in the temperate and polar ecosystems. They tend to be top predators, not necessarily apex predators in the systems they inhabit. And what makes pinnipeds really useful to study is that they have a terrestrial component to their life history, which makes them slightly more accessible than some of the oceanic-bound marine mammals, such as the cetaceans. But the coastal distributions of pinnipeds also means that they're coming into more frequent contact with humans through a variety of activities such as fishing, but also occurrence on beaches. You may have seen the images of people putting their children on sea lions, which is not necessarily a good thing for any parties involved. Because they're in these coastal ecosystems, they're a good indicator of what's going on locally, which is of concern to the coastal communities. So as I've put in the title of my talk, how did I go from studying pinniped diets to now aging dolphins here at Oregon State? So that's kind of what I want to tell you about. I'm going to start by talking about the end of my PhD research looking at gray seal diets on Cape Cod in Massachusetts. I then will be telling you about a project studying elephant seal demography in relation to foraging behavior that I did at UC Santa Cruz. And finally, I'll wrap up here by talking about the work I'm doing at OSU, aging cetaceans. So we'll start with a trip to the East Coast, looking at gray seals, which is in the image at right there. They're found across the North Atlantic, from about the mid-Atlantic states up through Canada and across the North Atlantic into the Baltic Sea. And across their range, we do know that gray seals tend to have a species-rich diet, and some common components across their range include sand lance or sand eel, flatfishes, gadids, and skates, among many others, with regional differences depending on where the seals are hauled out. And it's been assumed that a large portion of their diet can be attributed to commercially important fisheries species. In the US specifically, there's been concern about their predation on cod. And this presumed overconsumption of cod led to the formation of state-sponsored bounty programs in the New England region, which led to the near extirpation of gray seals from the New England states by the 1950s. With the implementation of the Marine Mammal Protection Act, and the end of those state-sponsored bounty programs, gray seals have slowly recovered, in part due to immigration from Canadian haul-out sites. And this increase in their population has been pretty consistent since the 1990s. But that being said, because they were gone for decades, especially when marine mammal science was really getting started, we don't know as much about gray seal ecology in New England as we do in other parts of their range. And their return to New England, particularly in the past decade or so, has reignited some of these concerns about gray seals and how they're interacting with a continually changing ecosystem in the North Atlantic. So how do you go about determining diet for marine mammals? There's several established approaches. You could attempt to visually identify the prey that are being consumed, but because most marine mammals are consuming their prey at depth and away from the coast, this doesn't really work for most species of marine mammal besides sea otter. An established approach is to identify prey remains such as fish otoliths or ear bones and cephalopod beaks that are found in tissues such as scat samples or from the stomachs of individuals that have stranded dead or bicaught. And this is great because you can identify prey from these diagnostic structures, and you can also enumerate them to get an idea of how much was consumed by the predator. But this method does tend to reflect short-term diet on the order of maybe a couple days, and it does tend to reflect coastal foraging. These hard remains also don't tell you if they were eating a larger prey item that may not have been consumed in its entirety, as well as the consumption of prey that lack any diagnostic prey remains, like some invertebrate taxa. So alternative methods have been developed and are being more increasingly used, one of which is DNA metabarcoding. So this is based on the amplification of small diagnostic fragments of DNA that tell you the species, hopefully species identity of the prey taxa that were being consumed. And this is great because it does increase the taxonomic resolution of the diet reconstructions that you generate, and it can identify prey that lack any diagnostic prey remains, but it also does tend to reflect shorter-term diet trends. And so one approach for longer-term diet trends is to use stable isotope analysis. In marine systems, we tend to use carbon and nitrogen stable isotopes. Carbon gives you an indication of the photosynthetic base at the bottom of the food web, and nitrogen gives you an indication of the trophic level of the prey, as well as the predators in the system. And while these stable isotope values themselves are not necessarily quantifiable, you can use the stable isotope values of the consumer and their prey in a mixing model framework to estimate the contributions of different prey resources to the consumer's diet. And while it's often suggested in the literature that you should be using multiple methods in tandem to get kind of the best comprehensive picture of diet of your consumer of interest, it's not always implemented. And when it is, it's usually the hard parts, which is the most numerical method and the most reliable in a way, with one that gives you either a greater taxonomic resolution, the DNA metabarcoding, or the isotopes, because it gives you potentially longer term information on the order of maybe weeks to months to years, depending on the tissues that you're sampling. And there's been a very recent trend of studies that are using 3 methods or more in tandem to give you a comprehensive picture of diet. And so that's what I did as part of one of my dissertation chapters, where we used the hard parts, the stable isotopes, and DNA metabarcoding to get a holistic picture about gray seal prey consumption in Cape Cod, Massachusetts. So by using these methods, not only was I able to reconstruct diet using each of these approaches, I was able to investigate some method-specific biases associated with each technique. We were also able to use the information generated from the hard parts and the DNA metabarcoding to see how that information may improve the estimation of prey contribution in stable isotope mixing models. And finally, we were able to investigate potential overlap between gray seal consumption and the fisheries resource use by commercial fishers in Massachusetts waters. And we're currently finishing up this work for submission to a journal soon. So we'll take a look here. This is the coast of Cape Cod, and each of those points is associated with one of these sampling sites where my collaborators collected seal scat samples from beaches that seals are known to haul out on. And these samples were collected between January 2016 and March 2017, so just over a year of information there. We did save a part of the fecal soft matrix, which is the fragrant part of the scat sample for use in the DNA metabarcoding and the stable isotope analyses. The remainder of the scat was cleaned and rinsed through a series of nested sieves to get all the prey remains that may have been in the scat sample. And we then enumerated these remains and identified them to the lowest taxonomic level possible. And from these different parts, I was able to calculate some metrics which are pretty standard to a diet study. We had a frequency of occurrence, which is a presence-absence metric, so how often does a prey taxon occur in the diet? We also calculated prey-specific number, which is an abundance metric based on the number of paired structures that are recovered from the scat samples. And then we calculated species richness, so the number of taxa that were identified in a sample, and the minimum number of individuals was estimated from either the paired number of left and right otoliths or upper and lower cephalopod beaks. Now, moving on to the DNA metabarcoding, one of the first problems we had to tackle was, were these scat samples from gray seals? In most of the range and for most of the sampling year that we had, they co-occur with harbor seals. So we first used a cytochrome B assay to amplify pinniped DNA and then optimize the PCR conditions in that reaction to only amplify gray seal DNA, so that we were confident that the samples that we were analyzing were from gray seals and not harbor seals that could confound our results. After that, we then used CO1, which is one of the standard DNA barcoding markers to identify prey. And we designed short primers to amplify fragments about 100 base pairs in length that would be able to diagnose fragments of prey associated with fishes, invertebrates, or birds, which are potential gray seal prey items throughout their range. The PCR products for the prey were sequenced at the University of Rhode Island's Genomics Center, and then bioinformatics processing through prey identification was accomplished in the CHIME2 software package. And from the sequencing data, we calculated a similar set of metrics. We again had frequency of occurrence, so that presence-absence metric. The relative read abundance was used to give a proxy of abundance. One of the drawbacks with DNA metabarcoding is you can't quite yet translate the number of sequence reads into a number or biomass of prey consumed, so this is a good proxy for right now. And then species richness was calculated in the same way, just the number of unique taxon associated with a sample. For the fecal stable isotope analysis, we first tested to see whether lipid correction was necessary, and we were able to do so mathematically. We did test it chemically. And we also had to do some sieving of the sample to separate the organic matter in the scat from sediments. Most of the beaches on Cape Cod are pretty sandy, so we wanted to make sure that inorganic carbon was not messing up the isotope values that we generated. We also, for the purpose of the stable isotope mixing model, had prey muscle from different species that are known or suspected to be gray seal prey that were collected as part of NOAA annual spring bottom trawl surveys. And we generated the stable isotope values from the fecal material and the prey muscle using a standard elemental analyzer mass spectrometer system. We then input all of these stable isotope values into a mixing model through the program MixSciR, and we generated 3 different model types. One had what's called an uninformative prior, so each of the prey resources that contributed to the model had an equally likely probability of contributing, and the other 2 were informed by the other diet metric or methods that we applied. So they were informed by the abundance data from either the hard parts or the DNA metabarcoding data. And finally here, we wanted to consider the potential for fisheries overlap with gray seal diet. So we obtained commercial fisheries data from Massachusetts because that's where the seals were foraging primarily from a website maintained by the National Marine Fisheries Service. And we generated 5-year averages to get an idea about average consumption in the years leading up to and including when the gray seal scat samples were collected. And we computed these averages for both landings in terms of pounds and for US dollars. And to calculate overlap, we use the Morrisita-Horn index. So this is a value that's generated that ranges between 0 and 1, with 0 meaning no overlap between the 2 consumers, in this case, seals and fisheries, and 1 being total overlap in resource use between seals and fisheries. And we calculated this index for both the frequency of occurrence data for hard parts and DNA barcoding, as well as the relative abundance metrics. So diving into some of the results here, the figure at right is a compositional bar plot. Each of the colors is associated with a prey taxon that was recovered from the hard parts data. On the left bar is the frequency of occurrence data. So again, that presence absence information. And on the right is that prey specific number, so the abundance. So what we found is that of the samples that were confirmed as originating from gray seals, about 1/3 of them were empty, which is a little bit unfortunate, but it's not unheard of that sometimes there just are no prey remains in the samples that you collect. But when we did find prey taxa, in general, there was just 1 taxon that was present in the fecal samples, which is a little bit lower than we would expect for a species that's supposedly eating many different species of prey. And so the species richness in samples was less than 1 on average. And when we look at the both the frequency of occurrence and the prey-specific number data, most of the contribution of prey was from sand lance, which is not too surprising because sand lance are an important prey type throughout their range. And I'll note here, if you look at the taxon identifiers, that most of these are not at the species level. They're associated with either genus or family. And this is because a lot of the hard parts that we did recover were damaged or eroded. And so that prevented the identification to a particular species. But this is why you use DNA metabarcoding to help get around that limitation. So when we applied the metabarcoding data, we found a much higher species richness in gray seal diet. So again, we have a compositional bar plot of gray seal diet with the frequency of occurrence on the left and the relative reed abundance on the right, and each color is associated with a different taxon. So you'll note that a lot of the identifications are to the species level, so that's one of the benefits of using this approach. We also identified many more species, so on average, species richness was about 7 taxa per sample. We also identified, of the 21 different prey we found, several new taxa that had not been previously recorded as gray seal prey in the New England region. And this included species like sea robin and common murre, which is a bird. But when we look at the relative reed abundance metric, most of the sequence reads that we generated were overwhelmingly attributed to sand lance. Again, not too surprising. And when we broke that down a little bit, we found that only 4 taxa had relative reed abundances over 1%, again, because most of this was overwhelmingly sand lance. Those other taxa included sea robin and flatfishes as frequently consumed prey taxa. And when we looked at the stable isotope mixing models, we found that, again, sand lance were a commonly attributed prey resource to gray seals. Again, not too unsurprising, but squid also popped out as an important prey resource to gray seals. So this is a density plot that was generated by MixSciR for the DNA-informed model. And what it's showing with each of the density curves is a different prey source in the model. The prey sources were squid, salmon, lance, mackerel, and a group of demersal fishes that were clustered based on their isotopic similarity. But we see overwhelmingly that most of that diet was attributed to salmon, lance, and squid. So when we take all this information together for the prey sources that were included in the stable isotope mixing model, we see some trends that are pretty consistent. Sand lance is a very important prey to gray seals, again, not too unsurprising. And relative to fisheries, it's not a very important fisheries target, and in fact was not fished in Massachusetts waters after 2014. In contrast, those demersal fishes that tend to be commercially important historically and today generate millions of dollars in revenue and millions of pounds of catch for fishers, but they tended to have variable contributions to gray seal diet. And when we calculated that Morosito-Horan index, we found that all of those values were less than 0.01, which indicates very minimal overlap between gray seal diet and commercial fisheries catches in Massachusetts, which is great news because it does kind of ease our concerns that there might be competition for limited resources in New England waters. So I hope I've showed you so far that multiple approaches will give you a generally consistent diet estimates, but there are some method-specific benefits to using each of these approaches. While the hard parts can give you the hard numerical data about how many were consumed, the DNA data will tell you more specifics about the different prey taxa that were consumed by gray seals. It was also reassuring to know that gray seal diet was composed largely of species with little to no commercial importance to fishers in Massachusetts waters. But one thing that we've considered and want to relay going forward is that while currently these commercially important fishery species are not a major part of gray seal diet, that doesn't mean that the predation pressure is not something to consider going forward when you're looking at ecosystem-based models that consider the effects of marine mammals on fishes and vice versa. Because marine mammals like seals tend to eat smaller individuals of these species, they may have a larger impact on younger age classes that have not yet recruited to the adult population. So it may be important to consider the impact they're having on particular life stages instead of just the adult population in general. So moving on to some of my research that I did at UC Santa Cruz, and it started with a seemingly simple question. Why are some moms so successful? This question was sparked by a paper that came out in 2019 looking at lifetime reproductive success of northern elephant seals. And the main results of that paper showed that a small number of females were disproportionately contributing to pup production in the colony of elephant seals at Año Nuevo, California, over many, many years. And when we generalize this, lifetime reproductive success is a metric that looks at the number of offspring produced over the lifespan of an individual. And so this reflects reproductive output and survival over time. And when we look at this over many different taxa, including terrestrial taxa, we see that this lifetime reproductive success is heterogeneous. So in general, there can be a wide variance in the number of offspring that are produced by an individual, and some will contribute more to subsequent generations than others. And when people try to look at why there's so much variance in the reproductive success of an organism, it often gets attributed to ecological luck. So just kind of random chance that some individuals are going to have more opportunities to reproduce and survive and contribute to subsequent generations. Now, others have considered pluck or individual quality. And when they do this, they try to relate behavioral strategies that may confer a fitness advantage that allows an individual to survive and reproduce more often than not. But what's important to consider is that some of these behavioral strategies may introduce trade-offs. So some behaviors may optimize reproductive output, but at the cost of survival and vice versa. There could be an alternative scenario too, where a behavior is used that optimizes both reproductive output and survival. And then the alternative to that is that there's no relationship, no trade-off, so to speak, because of a stochastic force, such as predation that may be random in essence. And we wanted to investigate this in particular because it's unknown how long-lived species like elephant seals are going to be able to adapt their behaviors relative to global climate change. So We worked with years of demographic, tracking, and diet data for northern elephant seals. They're probably one of the most well-studied species of pinniped in the world. You may know that they recovered from a near extinction event in the early 20th century, where the founder population may have been as small as 12 individuals, and they've recolonized most of their historical range. And because of this recovery, they've been monitored for decades now. And we made use of this long-term tracking data to answer questions about how foraging behaviors may impact the reproductive output and survival of female northern elephant seals. And because there's been these decades-long monitoring programs, we had some predictions about how these relationships may manifest. So we thought that mass gain on foraging trips would be associated with behaviors that maximize their mass gain. So they're foraging at certain depths and at distances from the coast that maximize their ability to find prey, that maximize their energy gain, which we thought would be associated with more fish in their diet. And we also thought that this would be associated with leaving the colony earlier to maximize the amount of time they have to forage at sea. We also thought that the pupping probability would be associated with an increased mass gain to allow for the survival of the mother as well as the pup that she is provisioning. We did not make any a priori predictions, though, about how mass gain and these behaviors may impact survival because we aren't really sure yet how those may manifest. There may be a trade-off between mass gain and survival because an individual may be foraging in an area that puts them at higher risk of predation. And if they get predated upon, they're not going to reproduce anymore. So as I've been talking about, we made use of a decades-long program at the Año Nuevo Reserve in California. As part of this monitoring program, elephant seals are flipper tagged with unique alphanumeric codes, as you can see in the top image there, and near daily resites between December and June, which is when the pupping season is, and then when they're on land to molt, means that we have a good indication about when an individual is born and when their last observation is, which is an indication usually of when that animal has died. We restricted our analyses to studies of 63 females that were tracked for 75 different trips between 2004 and 2015. We used this smaller subset because we were concerned about survival, and so we wanted to make sure that all the individuals had reached the end of their life so as not to bias the results of the survival analysis. And because of the long-term monitoring program, these near daily resites, we're able to calculate some demographic parameters, so we have a pretty good indication of their lifespan, their first observation usually as a pup to their final observation as an adult. And because we're focusing on mothers for this talk, we also have an indication of each instance that they were associated with a pup at some point during the reproductive season. And for a subset of individuals, we had diet data from blubber biopsies that were then analyzed for fatty acid analysis. And details of that are provided in a paper by my colleague, Chandra Getch. So we used a set of modeling techniques to look at these different behaviors. So again, these different behaviors were the mean depth of the individual when she was foraging, the departure date when she left the colony to continue her long foraging trip. Her diet was again based on those fatty acid data that were generated from the blubber biopsies, and her distance from the coast, so how far she was from the colony when she was foraging out at sea. We looked at correlations between each of these variables because some of those may be expected. We also used correlation analysis to look at how each of these behaviors may impact mass gain, so an outcome of an individual's foraging effort. We then used these 4 behavioral metrics and the foraging outcome in a generalized linear modeling framework to see how they may influence the probability of an individual successfully pupping or not. And we used a binomial distribution, so we modeled that as a 0-1, whether an individual was pregnant or not. And finally, for the survival analysis, we used a Cox proportional hazards model to estimate how these foraging behaviors or the foraging outcome may influence the probability of an animal surviving from one year to the next. So we'll start here with this group, the plots here. So scatter plots with the different behavioral metrics on the X and Y axes. So these are pairwise correlations. The trend lines and the associated confidence intervals are for statistically significant relationships, which are shown on the upper plots there. And so what we see here is that there were some significant relationships, again, ones we might expect, But when we look at mass gain, so we can focus here on the bottom left plot, that it was the only one that has statistically significant relationship with foraging depth, which again kind of makes sense. These individuals are probably maximizing where they can find their prey. And we were able to sample some individuals multiple times throughout their lifespan. And for these individuals, we were able to generate a repeatability metric. So how consistent or not consistent are their behaviors over time? And for those individuals, we found that they had good repeatability in these behaviors over time, which is useful. So if you only sample an individual once, you can get an indication of how they may be successful or not over time from a single sampling event. And in general here, this plot is a scatter plot of the lifespan of our individual elephant seals and their lifetime reproductive success, and they're colored by the mass gain of an individual, with the hotter colors being lower mass gain and the cooler colors being more mass gain. And we found a range of lifetime reproductive success for individual females from as little as 1 pup upwards to 16 with a median value of 7. And when we modeled these relationships, we found that mass gain was key to both pupping and survival. So if we look at the top plot, this is the modeled relationship between the probability of successfully pupping and the age-adjusted mass gain. Because individuals were tracked at a variety of ages, we did correct for age, so that was one less confounding factor here. So the curve is the modeled relationship between age-adjusted mass gain and pupping probability. The individual points are the individual seals. And we'll point out here that individuals that are the diamond, circle, and triangle, these were some that were sampled multiple times throughout their life. And we found that when they had more mass gained on a foraging trip, they were more likely to successfully produce a pup. So that was useful. And we found here that there was actually this sharp threshold that we found that below 205 kilograms gained on this foraging trip, an individual was not likely to successfully have a pup. And pupping almost always happened when they gained at least 260 kilograms of mass. And we also found that mass gain was important for survival. So the bottom plot here is the modeled results of survival at different ages, again, with age-adjusted mass gain on the X. And so what we see is that, again, mass gain, the more mass you gained, the more likely you were to survive from one year to the next. And this was important across different ages. And we'll point out here the bottom curve in that yellow-green color. That's the modeled probability of survival for an animal at age 15. Not many individuals make it to that age. And even at this older age, you know, they can gain more mass, and that's going to contribute to their longer-term survival. But because the survival probabilities are lower at this age, this provides an indication of senescence of this species, which is thought elephant seals don't really live longer than 2 decades. So this just kind of provides further evidence of that. And we did model the effects of these other behaviors, but they didn't have the same kind of statistically significant relationships like mass gain did on the probability of pupping or survival. And when we consider these together, we found what we're calling a physiological tipping point between mass gain, reproduction, and survival. So we'll walk through this figure briefly. So again, we have the 2 curves. The black one is the relationship between age-adjusted mass gain and the lifetime reproductive success. So the number of pups produced by an individual over her lifespan. The curve in red, sorry for those of you that are colorblind, is the probability of survival over time, again with age-adjusted mass gain. Each of the individual arrows are the percentiles associated with that number of mass gain. So what we find, again, is that there's this strong threshold relationship between the age-adjusted mass gain and the probability of successfully producing a pup and successfully surviving from one year to the next. And you can see that gains of hundreds of kilograms can make a huge difference in the ability for an individual to survive or reproduce. And we find increasing from 200 to 300 kilograms of mass gain on a foraging trip can contribute a 6-fold increase in the ability of an animal to produce pups, so that lifetime reproductive success increases from just over 1 pup over her lifespan upwards of 7. And we'll point out here on the curve that at the points where the 2 curves converge, an individual is almost always likely to produce a pup. And beyond that convergence point, Any continued effects of survival are due solely to having a longer lifespan. And below the convergence point, pupping is likely to fail, and an individual is likely to have lower survival over time. So taking all of this information in, we found that masking was critical for the reproduction and survival of female elephant seals. We did find that there were no apparent trade-offs between the different foraging behaviors that elephant seals were displaying on their foraging trips and their reproduction and survival over time. It's not too surprising that masking would have such a significant relationship because elephant seals are capital breeders, so they're gaining all of the energy that they need to support themselves and the successful reproduction of their pup on these long foraging trips after the molt. But what we were able to show here is how sharp this relationship is between masking, reproduction, and survival. And again, we'll note here that small changes in masking could have large implications for the persistence of the population in terms of reproductive output. And when we think about it another way, small decreases in masking could have population level consequences on the performance of individuals. There's been a couple recent studies of elephant seals that find that they're diving nearly continuously to feed to be able to maintain their performance to survive from one year to the next. So if things were to change, like for example, prey distributions, where the prey are occurring or what prey are available, that may reduce their masking. It may have negative consequences on the survival of individuals from one year to the next, and that could have cascading effects on the recovery or the continued recovery of that population in light of climate change. And the figure here is a conceptual model about how all of these behaviors relate between masking, reproduction, and survival, again, highlighting the importance of masking to reproduction and survival in elephant seals. So moving on to what I'm doing here at Oregon State. I'm studying aging in cetaceans, which seems kind of weird. I was telling someone a couple months ago when I was an undergrad, this didn't seem like something that was possible at the time. So it's kind of cool to be working on it now. And so age is an essential demographic parameter. It's important to know the age structure of a population to be able to see how it's increasing or decreasing and how that plays into the demographics of the population. But it's really difficult to determine without killing the individuals, which is not ideal. In cetaceans, which we're going to focus on now for this part of the talk, age can be determined by sectioning two different tissues. For your baleen whales or your mysticetes, this is done by collecting earplugs from dead whales. And in odontocetes or your toothed whales, you can section teeth, and you can count pairs of growth layers in either of these tissues to get an indication of the age of the animal. But again, because these are things that involve lethal sampling, this isn't an ideal approach when you're working with endangered cetaceans or those that are in recovery. So alternative approaches that minimize interactions and potentially and ideally don't kill the animals that you're studying are a necessary alternative. And one of these alternative avenues that's shown a lot of promise is the use of epigenetics. So this is a study of different things that modify gene expression without changing the underlying genetic code that you're studying. And one particular method that we've been working with and colleagues have been working with for a few years now is DNA methylation data. So what happens is the addition of a methyl group highlighted in the circle on the little diagram at right to cytosine. This can happen at other bases, but it does tend to occur at cytosine, which is one of your 4 nucleic acid bases in DNA, and typically in association with guanine. So we have these CPG sites is what they're called. And people have shown in organisms ranging from model animals like zebra and mice up to humans that there's a relationship between the chronological age of the individual that you're studying and the percent methylation that's occurring at these CPG sites. So they can either be strongly positively or negatively correlated with the age of an individual. And what we can do is use these methylation data in the construction of a mathematical model called a molecular clock. And like Scott mentioned in his introduction, the species that I'm working with are Cook Inlet beluga and Hector's and Māui dolphins in New Zealand. I won't be talking too much about beluga today, but we are working with samples collected from the Cook Inlet distinct population segment, or in Alaska. So this is a genetically distinct group of beluga that are on a continuing downward trajectory. And Ellie Boers, a former postdoc here at Oregon State, developed a species-specific clock for beluga. And what I'll be working with are some additional samples we've collected since that clock was published to refine it a little bit more, but also to generate some data that could be useful for informing management and conservation of Cook Inlet beluga with our partners at the National Marine Mammal Lab in Seattle. But mostly what I'm working on is developing a molecular clock for Hector's and Māui dolphins in New Zealand. They're endemic, and Māui dolphins in particular are endangered right now. And it's thought that bycatch is the main threat to the persistence of this species in New Zealand, but there may be other things to consider like toxoplasmosis. And so the goal of the project is to develop a species-specific clock for Hector's and Māui dolphins to estimate age of the population that's existing and see if we can help inform management and conservation of this species. So there's a lot that goes into constructing a molecular clock, and I won't go into all the details here, but this is kind of a quick, broad overview. So we first have to start with genomic DNA, which we got from skin samples. We're working with two different data sets here. To calibrate the clock and construct the model, we have paired skin and tooth samples from dolphins that were stranded along the coasts of New Zealand, and we extracted genomic DNA from that. We also have biopsy samples that we're currently analyzing right now to estimate those ages later. But once we get the genomic DNA, we then use a bisulfite conversion to allow for the methylation data to be generated. These methylation data are then read on an array technology that was developed by collaborators at UCLA. And it's a custom array that measures methylation at over 30,000 sites in the genome of the animals that you are studying. These data are generated with p-values that are indication of the confidence in the methylation estimate for the amount of methylation at each of the CPG sites. And so we're using these p-values to kind of help refine the clock by removing sites that do not have a significant relationship with our known age individuals. And again, these individuals have their known age based on counts of tooth growth layer groups. Now, from the CPG sites that have significant relationships with age, we're then using those as potential terms in these mathematical models to estimate the relationship between the amount of methylation in the genome and the age of these known age individuals. And we're using an R package called GLMNet, which is generating essentially a multivariate linear model that's penalized so it's not over parameterized, and it allows many different sites to drop to 0, so they don't really matter in the model, while optimizing a smaller subset of sites that do have a relationship between age and methylation. And once we finish calibrating the clock, we can then apply it to our other set of data, these biopsy samples from living Māui dolphins in New Zealand. And since I've only been here a couple months, we aren't done with this yet, but we do have some preliminary results that we'd like to share with you today. So for these known age individuals, known being because there's a little bit of uncertainty in the age estimates that are being generated, the age range we have for individuals is between just over, or born to about 14 years of age. And for the genus, the expected longevity is about 20 years. So we're missing some older individuals, but this is a pretty decent age range. And we have 19 females and 12 males in this set of known age individuals. And the histogram at right here presents the absolute values of correlations between the amount of methylation at CPG sites and the ages of these known age individuals. And we find that most of the CPG sites do not have a strong relationship between the amount of methylation and the age of the animal. And that's to be expected, but we do have at least 4,000 sites that do have some relationship between age and methylation. And so we can use the smaller subset of CPG sites to construct the final clock models. So stand by for more results in the future. And then here is just a couple of the CPG sites that have a strong relationship between age and methylation. So the panel on top is the CPG site with the single greatest positive correlation, and the bottom is the single greatest negative correlation between age on the x-axis and methylation on the y. And so when those relationships explain about 80% of variance in the data, so that's pretty promising for the development of these clock models going forward. So to kind of wrap up here, some of the conclusions from the research part of this talk, I hope I've shown you that by using complementary approaches to study the diet of pinnipeds, we're able to generate a pretty consistent diet estimate, and we were able to find that while gray seals have been accused of eating a lot of commercially important fishery species, we instead found that their diet is mostly composed of species with little to no commercial value. So this will be useful going forward because we've helped to contribute to a new baseline for seal diet habits in the New England region. And this is going to help inform future studies that are looking at energetics of seals going forward, and to help inform those ecosystem models that will help give a better indication about the state of the North Atlantic ecosystem and how different organisms are going to respond to climate change going forward. I also hope that I've shown you that small changes in mass gain and foraging effort can have large consequences for the survival and reproductive output for female northern elephant seals. And this again, this kind of information will help guide management and conservation of the species going forward, again, in light of global change. And finally, the methylation data that we have generated so far shows a lot of promise for developing a clock to age Hector's and Māui dolphin. And I wanted to end here with some kind of career conclusions. So how have I gone from studying pinniped diets in grad school to aging dolphins here at Oregon State? The first thing that I thought of was that I've been able to hone skills and apply them in new areas. So prior to starting here at OSU, I've never done anything with epigenetics before, but I have years of experience working with DNA metabarcoding data. And by working with molecular data like that, I was able to kind of easily jump into working with a new type of data in a new way. And also, as I've gone from different grad program to grad program to here now doing my second postdoc, I've been able to kind of expand conceptually what I'm thinking about. So my work on diet has helped influence how I look at foraging behavior and how that ties into demography and population ecology. And that's impacting the work I'm doing here. And networking has made a world of difference. This is probably cliche and not too surprising, but I'll point here this map. I made this earlier this year when I was giving a talk to some high school students about my journey in science so far. And you'll see that I've been able to go kind of all over the country studying science, which is kind of insane. And I've been really fortunate that I've been able to leverage some connections that I've made over time to kind of keep moving forward and not fall out of the leaky pipeline that sometimes happens in academia. I'll highlight the line that goes from Hawaii to Massachusetts. That was a job that I was offered and accepted after undergrad because I did an internship with a person at National Marine Fisheries Service in Woods Hole. And that went so well that she offered me a job after my undergrad was complete. And that job led into meeting my grad school advisor at Māui Marine Labs. But also I was able to interact with scientists in Woods Hole who would then become my collaborators during my PhD research on gray seals. So even for an introvert like me who doesn't socialize a lot, making at least a good impression is useful down the road. And finally here, this is a quote that I got from one of my postdoc advisors at Santa Cruz, that you miss out on 100% of the opportunities that you don't take. If I hadn't listened to Carlos at that point in time, I would not be here today because I was having doubts that I could apply what I had learned to an epigenetics framework. But I applied and now I'm here studying epigenetics at Oregon State. So that being said, don't disregard an opportunity because you may not be 100% qualified for it. You know, even if there's an opportunity where you don't get it, maybe something else will fall in your lap. And that's what happened with my PhD. I reached out to my advisor at Louisiana State and for a project that was going to study penguins and seals in Antarctica. And it sounded super cool. I've always wanted to go to Antarctica. I didn't get that project, but we were able to look at my CV and his history and found that we had a shared connection interested in seals in Woods Hole and in Massachusetts in general. And so we leveraged that connection that we had and with our collaborators that we knew, and that resulted in a really cool PhD where I was studying gray seal foraging ecology. So there's always an alternative path that could work out for the better. So I'll end here by acknowledging all the collaborators that have made the science that I've been doing the past few years possible. Lots of collaborators and the different funding agencies that have made all this possible. And with that, I'd be happy to take any questions. Thank you.
Great presentation. Do we have questions? Yes.
Fantastic presentation and really, really great research. I'm a little bit curious about the one of the things that you said early on about gray seals is that they were accused of eating cod.
Yeah.
And my recollection, and you can please correct me if I'm wrong, is that there has been some change in the distribution of cod in recent years. And
Right.
because of the accusations being made before they recovered or were depleted, I wonder about the conclusions that they are not greatly eating a lot of cod. Perhaps the distribution is so much different today. So I guess that's kind of the one question. Also for fisheries, I saw that you saw a lot of squid, which are of commercial value, not like sand lance. And obviously it's a relatively small proportion in terms of, overall catch and overall ex-vessel value, which I think was your metric. But for individual fishers, typically squid are caught in what are often called coastal pelagic fisheries with purse seine nets. And some of those vessels are fairly specific in the types of fisheries that they're able to participate in and prosecute. And so it may be perhaps a greater problem for those particular fishers than for fisheries in general, in the big picture. So
yeah, those are great questions. So in case anyone didn't hear, the first question was about the distribution of cod having changed recently, and that's true. The North Atlantic in particular is experiencing some of the fastest warming in the world right now. And because cod are kind of a cold water obligate species, they may be shifting their distribution northwards. And so gray seals may not have opportunities to eat as much cod as they may have historically. And that could be very true. As I've shown in the presentation, they didn't eat much cod that we found. And again, that could be because, like you suggested, they don't have cod available to eat. But that may be less of the case for seals that are foraging in Canadian waters. The cod may be shifting their distribution northward into Canadian waters, and so seals that are foraging in Canada may be continuing to eat cod. But in New England, it may be a little bit different. And that could be possibly what's going on here. Some of the species that I recovered from the metabarcoding data in particular are species that may be vagrants that have been brought in by the Gulf Stream, and other species that may be associated with warmer waters, more associated with kind of Long Island Sound and more mid-Atlantic waters than those offshore of Cape Cod and into the Gulf Stream. So there was that question. And then about squid, yeah. So individual fishers and fisheries may be more suspect to interactions with gray seals, and that's true. I didn't talk about it today, but during my PhD, I was involved with a group called the Northwest Atlantic Seal Research Consortium. So they're trying, it's bringing together scientists, managers, and fishermen to try to talk about seals and guide research going forward to kind of come to the best solutions for everyone involved. And one of the things that they are doing is trying to optimize gear in a way that minimizes interactions between seals and fishers so that kind of everyone involved gets the best bet. The seals can eat, the fishermen can fish, and they won't get shut down. So yeah, so squid fishers in particular may be subject to more interactions with seals. That's true. I did show in the talk, seals have learned how to jump into weirs and take advantage of that. Essentially, they have a buffet that they can take advantage of for an easy meal with minimal effort besides jumping in and out of a pen. So yeah, that is something to consider going forward. So many options.
Thank you. That was a wonderful talk. It was fascinating. 2 questions, mainly about the epigenetics.
Okay. So
does there have to be a unique, essentially, aging graph for each species?
There doesn't have to be. There have been clocks that have been published that work with a group of species. One of our collaborators at UCLA has been involved in the development of a Panodontaceae clock. So that worked with tissue samples from a variety of toothed whales to develop one that was more generalizable to all toothed whales instead of species specific. But you may want to consider a species specific clock so that you can get the best estimates possible.
Secondly, can those clocks be applied to species with longer age ranges, such as whales or perhaps orca?
Yeah, definitely. So there was recent work that has used a smaller number of those CPG sites to age humpback whales, which are going to be living much longer than these 20-year-old dolphins. And also epigenetic clocks have been developed for organisms as long-lived as elephants. So those are living, nearly a century. And so the epigenetic approach with the DNA methylation data shows a lot of promise for organisms that are really short-lived to those that are living centuries potentially. And even some of the human studies are working with people living over 100 years. So there's a really good age range, especially for the human studies so far.
Fascinating. Thank you.
That's a great presentation. Thank you.
Thanks.
Greg stole my question, kind of, is that I guess when you're looking at the gray seals and the elephant seals in your study, it has a very discrete distribution relative to the things that they might be preying on.
Right.
And so when you find out that the gray seals on the East Coast are eating a small proportion of the commercially important things, of course, there's always the concern that, well, what are the rest of them doing?
Mhm.
And then, but is there, and do you have any idea how well their diets correlate with what they are eating in, say, biomass distributions? Do they actually live where they need sand lands? Is that what's going on?
Yeah, so that was a question about distribution of seals overlapping with their prey. So yes, there hasn't been a lot of tagging work on gray seals in New England yet, but a study did show that they are kind of generally foraging on these offshore banks that are close to the coast in Massachusetts in particular. So that would expose them to habitats where you're going to find a lot of sand lance and these other species that we are pretty confident that they're going to eat. So some of these flatfishes and some other schooling prey like herring. And these prey are also are consumed by other predators like seabirds and whales and whatnot. So yeah, so they are eating generally where they live. They're not, gray seals are not undertaking these kind of long distance migrations like elephant seals do, where they're going, thousands of miles out to sea to forage in, mid-water habitats, where they're going after a totally different prey type than what would be found in coastal California.
Thanks so much, Keith. That was a really fantastic talk. I have a question about your elephant seal study.
Sure.
It was interesting to me that diet wasn't really, or it didn't seem to be that much of a driver or predictor.
Mhm.
And so you had diet data. So did the elephant seals just have a very uniform sort of diet, or what do you think was driving that?
Yeah, so that was a question about elephant seal diet and what may be driving the lack of relationship that we observed. So I think one important thing to keep in mind is we did only have the diet data for a subset of the females that we tracked over time. So, that is going to limit the inference that we can make. But also, yeah, it may come down to it's less important what they're consuming versus how much they ultimately gain. And I think that's what we've showed with the mass gain data is they need to gain a certain amount of mass to be able to provision themselves, and then for the females in particular, make sure that they have enough to provision their pup. So it may not matter at the end of the day whether they're going after a whole bunch of fish or a whole bunch of squid. We did think that they were squid obligates for most of their time, but recent work has found that they're eating more mesopelagic fishes too. So that's kind of what we thought is maybe because we recently found out that fishes are more important, that maybe fish specialists would have a better reproductive output, and that wasn't the case. But you never know what could happen with more samples down the road.
Thank you, Keith. That was a really nice presentation. Another elephant seal question.
Okay.
And it's if you could elaborate a little on a couple of the covariates that you used, distance from the colony and depth of dive. I'm presuming that you're using the farthest out distance for the trip, and also maybe either the maximum depth of dive or the last dive or something like that. And what I'm getting at is it appeared to me that the implication was that animals going farther away from the colony and also diving deeper were showing higher mass gains. Is that correct?
Yeah, so this is a question about the covariates that were used in the elephant seal analyses. So yeah, what we ended up using was the average dive depth on these foraging trips as opposed to maximum dive depth. And we used the median distance from the coast as opposed to kind of the maximum trip distance. And that's kind of been the standard suite of metrics that have been used for these elephant seal studies for a while. So we kind of kept that for consistency's sake. But yeah, you're right. We did think that individuals that were foraging further out to sea are diving at deeper depths. And yeah, that is what we saw there. And they are kind of closely correlated with each other too. So we did kind of keep that in mind for the analyses that we presented.
Thank you.
Hey, I've got two for you.
Okay.
So the first is about gray seal diets.
All right.
And I was curious, I know with time constraints, we always have to pick the metrics that we estimate, but did you get into proportions of prey by weights? Essentially my question is because I think I'm curious if your relative reed abundance
Right.
would be more comparable to measures of mass than by number. And then also with your stable isotope work, all of a sudden squid came up as super important. And
Mhm.
if you were to estimate proportions of squid by weight and convert from, the size of the beak to the expected mass.
Right.
Did you do any of that to see if those were related?
Yeah, so this was a question about whether we looked at metrics by weight as opposed to just number. And we didn't end up doing that because most of those hard parts were damaged. And so it would limit the ability of us to estimate the mass of the individual prey items that were consumed by gray seals. So it's something that we wanted to do. I did that as part of my master's work, where we were able to reconstruct the masses as well as the size of the different prey types that were consumed. But we weren't able to do that because most of the structures were too eroded and damaged to be able to apply the appropriate regression equations to estimate the weight of the individuals. We were able to do it for the size. So we were able to do that, but we weren't able to do that for the weight. But yeah, it could be something to investigate in the future, whether weight would have an impact on the reconstructions that you get for those stable isotope mixing models. And the other thing to consider with those models is that a lot of those prey are feeding on kind of the same base organisms, so they overlap a lot in isotopic space. I didn't show it here, but all of those demersal fishes, especially a lot of the flatfishes and the gaddids, they are pretty much on top of each other in bivariate isotopic space. And so it's kind of tricky to tease apart whether one organism is driving that or not, because they're pretty much similar isotopically. So that makes it a little tricky. So yeah, it could be that squids stood out because they are isotopically distinct in the same way that sand lance are isotopically distinct from the other prey types. So it's just something to consider. And maybe stable isotopes isn't the optimal method for that particular system, but it does have applicability elsewhere.
Awesome, thanks. And if I
Yeah.
can do one more.
Okay.
For your aging of dolphins, I was curious how solid of an assumption it is that the ages you get from the teeth are accurate ages. Is there enough information? Has there been any validation on that to say, yes, that is the truth? In the fish world, we use physical or chemical tags.
Right. Yeah. So it's been something that we'd be considering going forward. But yeah, in general, it's generally agreed upon that a pair of growth layers is about 1 year of growth for that individual. But teeth can wear down. And so some of those, the older age layers may be more difficult to ascertain from the tooth sections that are going forward. We've also had issues where if the centroid is missing from the tooth, so kind of the pulp cavity that gives you like the base age 0, if that's gone, it does make it a little tricky to estimate whether you're looking at just growth layers or the centroid plus growth layers. So that introduces a little bit of uncertainty into the ages that you are using to calibrate the model. So I didn't present it here, but we are working with kind of 2 calibration sets and seeing which is performing better. We have what we're calling a strict subset of those individuals with kind of the best tooth age estimates, like there's really clear images with the centroid and the layers are really distinct, versus some that expand our age range, but may introduce some uncertainty in the tooth age estimates. So we're seeing how that's going to impact model performance.
Any other questions? I think we're at the end of our time. Everyone, would you please join me in thanking Keith for her great presentation. Thank you for joining us. Grab a cookie on the way out.